Yeah, so this is how to calculate the study days. You know, right, the dose given day also we should consider as a 1 day. Okay, so I mean, when calculating the study days, we need to remember that one also, how to calculate studies. Let me show you a small example. Here 5th January hemoglobin test conducted, and the value is showing 8 grams, 8.0 grams. It's some laboratory value of the hemoglobin. On 5th January we conduct the test. Then 6th January we have given the dose. So whenever you give the dose, so that day will be considered as the first day. Then 15th January the patient visited first time. Means on which day the patient visited. Date is 15th January. So today minus dose given date. So 15 minus 6. So it will be 9. Plus 1. We have to add 1. Because the 6th day also we should add. 6th January also we should add. So it will be the 10th. So 10th day was the first visit of the patient. Then the study completed on 30th Jan. Means how many days the study duration was, means 30 minus 6, 24 plus 1. So 25 days is the study duration. Okay. So this is how we are calculating the study days. You know, there are different ways to calculate the study days. See, dosing day as 1 day, I mean first day. Then study day equal to present date, it's not day, present date minus dosing date plus 1. Okay, so it will give the study days. Then here we have a small example. You know the INTCK function. So using this INTCK function, we can calculate the study days, and we are adding plus 1. Okay, so 6th January, then 30th January. So 30 minus 6, which will be 24 plus 1. So it will give the 25 days. So you can use the INTCK function to calculate or to find out the days between the two given dates. Okay, or else if you have multiple things like this, it's a dose, means it's a dose day, is a TTE, means treatment end date. So how many days the duration was. So this minus this, you know the double trailing, right? Double trailing means it will consider as this as a dose day, this is a treatment end date, and treatment end minus dose date plus 1. Okay, so anyway it will give the study duration. Okay, like this. Then I mean, this is simply calculating the study days. Now let us see the time to event or time to sensor. So what do you mean by time to event or time to sensor? Okay, here I will show you a small example. Time to sensor, time to event means actually when somebody like participate in a clinical trial, for example, a patient having some seizures, okay, or some fits, okay, or some epilepsy, some type of disease. So we started I mean the treatment to that patient for a 6 months of period. Let us say in the 6 months of period, so whether he exhibited that epilepsy, means any episode happened in that trial period, so that he had to write. So it's always, it is a binomial value. Binomial value means whether it happened or not, only two. So heart attack, whether it happened or not, heart attack or no heart attack, death or no death, seizure or no death, no seizure. So we have only the binomial values, yes or no, that's it. Okay. So after this you will understand what is the time to event or time to sensor. So there are 4 patients. So usually patients we call them as the subjects in this clinical trial. Okay, so 4 subjects they enrolled in the clinical trial. Okay, on 20th, on 3rd January. So at the time of joining, the 4 patients, 1, 2, 3, 4, the 4 patients' status was showing very serious, means their condition was so critical, because they are not good. So at the time of joining, all the 4 patients joined on the same date, 3rd day. Dose given to the patient on 3rd day, 3rd January only, and on 20th January, death reported for 101 patient. Death reported on 20th January. So we made a note of this one, right? Death happened to the 101 patient on 20th January. Then out of curiosity, then we thought to know the information of 102 patient also. Okay, then 102 patient was showing like no death happened to that 102 patient. Means we got the information of 102 patient on the same day, 20th January only. But later on, when we tried to contact them, then he was not available. Means last known information about that 102 patient was 20th January only. Then coming to the 103 patient, so he took the treatment, okay, and he was there Okay, he was there till end of the study. So when the study ended, study completion date is 31st January. So study started on 3rd January, study completion day is 31st January. So 31 minus 3, it will be 28. 28 plus 1. Okay, so 29 days. So the total study duration is 29 days. So the patient was available the 29 days also. Okay, means we know the information about the 103 patient. Then 104 patient, he took the treatment and he disappeared. Means when we tried to contact him at the end of the trial or in between also, he was not available. So this is the history of the 4 patients. Okay. Then what is the time to event of the first patient? So what's the time here? I mean, when the death happened to the patient, 20th January. Means how many days? Means 3rd January he took the medicine, then 20th January death happened means 20 minus 3, 17 plus 8, 1, 8. So 18th day the death happened to the patient. Okay, 18th day, right? So here the time is the day and event is the death. So we call it as the 18th is the days to death. Days to death here. Time to event for 100 subject is event date minus dose date plus 1. So 20th death happened, 20 minus 3 plus 1. So 18th. So 18th day, we call this 18th day as days to death is 18. So 101 patient, days to death is 18. So you should spell like this, days to death is 18. Then what about this 102 patient? 102 patient, he disappeared, right? So last known information about that 102 patient is 20. Okay, so time to censor for, okay, what about this 102 patient? Time to censor for 102 patient is, so why we are calling it a censor? Censor means no death happened. So censor is stopping, right? Stopping. So the patient information we got up to 18th day only. We know the last known information about the patient is 18th day only. So time to censor. What is the time here? Days. So days to censor for that patient is 18. So for first patient, days to death 18. For second patient, days to censor is 18. Then third patient, no event happened means he was there. So 31 minus 3. What is the 31 minus 3? It is 29. Okay, 31 minus plus 29. So what's the 29? 29 is the days to censor. Days to censor is 29. Then what about 104 patient? No, nothing is available. So what we do? We make it as a missing only. So we don't give anything here. So 104 is missing. No time to event, no time to censor. Okay, no days to censor or event. Okay. So this is how we are calculating the time to event and time to censor. Okay. Now let us take this example. Here, okay. So we should have 3 data sets. Why? One is dose given date we should have. Second is when it ended. Then whether event happened to the patient or not. So we need 3 data sets. Okay. So we have the first data set. This is dose date, subject. Then it is end of study, because the study may not start for all the patients on a single day. But here, just as an example, we have taken the study day for all the patients is same, but it may not be the correct one. Okay. So here, this is a dose day. So for 101 patient, we have given the dose on 1st January. For 102, given the dose on 3rd January. For 103, 6th January. For 104, 9th January. So we have given the dose to different patients on different dates. And this is the end of study. Because when the dose day is different, then the study day also it's different, right? I mean the end of the study also it's different. Then whether event happened or not. So it is about the event. So this is the event. Dose date, end of study, then event. So here what is the event? Seizure. 101 patient, 1 means yes, 0 means no. So 101 patient seizure happened. So how to calculate the time, I mean the days to seizure for 101 patient? Seizure happened on 5th May when he joined 1st January. So 5th May minus 1st January plus 1 is considered as the days to seizure. Then for 102 patient, no seizure happened, means here we have the information that no seizure happened. So no seizure happened means there won't be any date, right? If it happens, then when it happened. When there is no seizure happened means So how can you say that no seizure happened? Because he was there till at the end of the study. Okay. So no seizure happened means end of study minus dose date plus 1. It will give the days to sensor for the second patient. Then 103 is missing. Missing means we don't have any information this 103. So it will be missing only. But about 104, event happened. So event happened date minus study date, dose date plus 1. It will give the time to event, right? Okay, let us see. First what we do, we merge all the three data sets, right? So merge all the three data sets. Okay. Then based on the condition, and also we'll be creating a new variable like this. See this output. Now 101 patient is, yes, seizure happened. On which day? 126. What is this 126? Days to seizure 126. 0 means it's a days to sensor 221. So third one is missing, no data. Fourth one is days to seizure. Days to seizure for the fourth patient is 151. So like this we have to calculate the study days. Okay, when the event happened, then how many at what day event happened. So it's not only days to seizure. Someday they will ask you to calculate the months also, days also, weeks also, whatever the duration may be, time to event. The time may be days, months, weeks, hours, whatever it may be. Then event means what is the event, whether it is seizure or death or any event, we can write like this. So it is just a small example of time to event. Okay, so this is one small example. Okay, now let us see how to create export transport files. What is the use of export transport file creation? Okay, so when sending the data sets to the FDA, while sending the data sets to the FDA, we won't be sending the data sets simply like this. See, for example, we have so many data sets in my system. See the data, I have so many data sets, right? We should not send the data sets like this to the FDA, US FDA. We should convert them into the export transport file. See, right click, go to the properties. It is showing us .SAS 7b dot. So it's a SAS extension. It's a data set extension, but you have to convert it into the export transport file. So export transport file will compress the data set. Okay. So anyway, we'll be sending in the export transport file only. When you are sending the the data sets directly, they won't accept. Okay, we should convert them into export transport file. Let us see how to convert into export transport file. You know the library engines. Okay, so I think you might be knowing about the library engines like Access, Excel. So we have different types of the library engines. So in the same way, we have to create one library engine. So first I am selecting a location. I am creating a new folder, D drive Rahul. Okay. In my D drive, I am creating a new folder with the name called the Rahul. Okay. In this, first I want to store this data. Okay, end of study. Okay, so this is the end of study. I saved it in the Rahul location. And how to convert into the export transport file. Okay. So okay, let me write it here using the library engine. So what is this Apple? Apple is a base engine. Okay. Now let's create one export engine. Export engine means lib name, some orange. Export means export engine we are creating. Give the location, D drive, Rahul. Give a name. Okay, some EOS.xpt. Okay, means we are creating a new location. What is this? Orange. What the orange is showing? Orange is showing this location. D drive Rahul EOS.xpt. Okay. So we created a export library. Then from the Apple EOS copy into the orange like this. Data orange dot EOS set apple.eos run. So I'm copying the EOS which is present in the Apple to orange.eos. Submit it. Got copied. Now go to the Rahul. You can see the EOS. It is showing as a SAS file, but it's not a SAS file. See, right click and go to the properties. It is showing the export transport file. It's a .xpd file. Okay, it's a .xpd file. So you have to convert into the, actually the XPT extract, it should not show like this. There is some error in my system. Okay, it shows differently. No, here the look will be different. One second, let me show you in the internet. I don't know whether you can open in your system or not. Export transport file. Yeah, it looks like this. You know, this one. The export transport file, it looks like this. The icon will look like this. In my system I have some problem with the extension, but it looks like this. Okay, so the icon will look like this. Export transport file. Okay, so what we have done, we converted our data set to export transport file. Right click on the data and see the size. It is showing 5 KB, some 8 KB, but when you open it, it is showing 1.09 KB only. Instead of 5 KB, it is showing 1.09. Means it compressed a lot. So it is like zipping the files. Okay, but whenever you are sending, each dataset should be converted into each export transport file. Actually, in one transport file you can keep multiple datasets also. It is just like a suitcase, right? Suitcase or some briefcase or some, you know, some bag. So you can see here it is showing like this, right? It's a bag. It's like a bag showing one dataset. Inside there is one dataset. Means in one bag there is a dataset, but in one bag you can keep multiple datasets also, like this. But US FDA, it's not recommended. You should keep only one dataset in one transport file. See here I kept this UOS in the orange, right? Then data orange.class, set SAS help.class, run. So we are copying the class also, which is from the, which is in the SAS help, to the orange. Now open this orange. Orange contains the class, okay, but it replaced, right? Overwritten. It replaced. Then okay, let us see. Proc copy in equal to orange. So into the orange I am copying. No. In means in the apple. Out equal to orange. Select US, run. So open the orange. Okay, I think, yeah, it's overwriting, right? I think, you know, usually here it is showing we can't copy two datasets into one export transport file, but earlier we did, right? Orange.us, okay, into the orange, we copied. So writing, it seems. Open. Now orange is showing only class. When you copy the US also, it is showing in the place of yeah, I think it's holding only one dataset. But in the, I used the earlier versions, you know, us.xpt.
Kiran?
Yeah.
You know, data orange.us and set apple.us. Are you actually creating, or you are copying the data US from Apple to Orange?
Copying only, right? We are copying the US, okay, from the apple to orange. So orange is a library. Here you can see the US. It contains the data, but you cannot open it because unless you convert into a dataset, we cannot open. Got it. So this is how we, okay. Let me show you again to avoid the confusion.
I'm down here. Yeah.
No, yeah, I will show you again to avoid the confusion. See, somebody gave us the data sets like this. One second. Okay. So see, these are not the data sets. These are export transport files only. See the type showing AE SAS export transport file. Then it is a SAS export transport file. So these are all the export transport files only. So how to, let us assume somebody gave us this export transport file and you want to see the contents of this AE. How to see the contents of this AE? First we'll open the SAS. Now after opening it, this is the location where the export transport files are there. So libname, give any name, okay, some alpha, okay, so give any name alpha. Then we are creating an export library. Then where is the location? So this is the location where your files are. But you're opening this particular AE, right? So AE.xpt, give the extension and open. So when you create a library, then it will show you alpha with a different globe mark. Why? Because it indicates that it is a export transport file library. Then right click and go to the properties. It is showing the location and engine also export. Then when you open it, it will show the AE is present. Now AE is the data set. We cannot open it here. See, it is showing because we cannot open the data set which is present in the export transport file. Then we have to copy it into some other location. So how to copy? Data work.ae set alpha.ae. So we are copying from alpha to the work library with the same name, submit. Then it got copied into the work library. Now you can open it as a data set. You can see there is the data here. There are only 16 observations and we have some data here. Okay, like this we are converting the export transport file into data set to view the contents. Okay. So in the same way, if you want to convert this again into the export transport file, then use the same method. Give a location, okay, and then copy from this. Okay, okay, I'm deleting this. No, I'm also deleting this alpha. Nothing is there. Now in the work library, I have this AE. I want to convert this AE into the export transport file. Okay, so you select any location. Okay, let us remove this. So I am selecting this data raw location to create the export transport file. Okay, then libname. So give any name. Okay, some clinic. Then export library we are creating and give the location where you are storing and a.xpt. Submit. After submitting, then it will create a folder called the clinic giving this location. Then from our work library to clinic, you just copy using either copy or set data clinic.ae set work.ae. So we are copying the work from AE from work to the clinic. Submit it. Okay, got copied into the clinic. Means it, whether it is it's in the clinic means it created the data set in this location. See, in the Rahul we have this AE which is a export transport file. Clear? So this is the way to convert the SAS data sets to the export transport file. Clear? So yeah, this is how we will be doing. Got it? But actually there are different ways to convert the export transport files into data sets or data sets into the export transport files. Okay, let me show you a small example here. When you open the SAS, the US FDA website, okay, export transport file US FDA. We can find so many papers here. See this study data specifications. Okay, Study data specification means whenever you are sending the data sets to the FDA, how you will be sending the data sets to the FDA. So these specifications are there here. Now, see, data tabulations. The tabulation is nothing but the data sets. So SDTM data sets and analysis data sets. So whatever it may be. Yeah, SDTM or ADAM. Then see, the submission of non-standardized data sets is not recommended. If provided, each dataset is provided as a SAS transport file. There are no further specifications for organizing legacy datasets. Okay, so you should send in this format only. Okay, and one more thing here. Yeah. So anyway, you know this organization, how you will be sending datasets, then study analysis, ADAM legacy. So I mean, anyway, you will understand this, how you are organizing to send to the FDA, the listings, all those specifications. These are study data specifications for posting or submissions. Actually, I am not USFDA seaport or seaport. Yeah, see this. Submit the clinical trials datasets to FDA using the right XPT file format. When we submit the clinical trial datasets to FDA, we need to convert the datasets in original format to XPT format. There are two ways to convert the SAS datasets to XPT format. However, only the .xpt format generated with proc copy can be accepted by FDA. So here, you know, what we have done, we just copied. See, you can use either set or copy also. So data clinic.ae or else proc copy in from where work library out into clinic select. So which data set? AE. So this is called the copy. So you can do like this. See, I am deleting this AE. Okay, clinic, right? Okay, let's delete the clinic and create a library and then run it. So proc copy also will do the same thing. Okay, either you can use the set or proc copy. It is recommended. Okay, proc copy can be accepted. In FDA study data specifications section 2, this is what just now we saw, study data specifications section 2. Okay, here the one which we saw now. So in the study data specifications is very specific about the data format that FDA will accept. Okay, it emphasizes that SAS transport file processed with the seaport SAS proc cannot be processed or achieved by the FDA. We can do using the seaport technique also. Means there is a separate procedure called the proc seaport. So using this proc seaport also we can convert into export transport file. But that is not accepted by the FDA. Okay, so it is all about the export transport file only. Okay, you just check this blog also. It's a blog. Okay, now. Oh yeah, but you should know how to, you know, use the seaport and simport also. Actually, I will do one thing. I will just send you a video, okay, about export transport files, because it's not at all related to the clinical. If you can listen to that video, you will understand, I mean, little bit about what is the export transport file creation. So yeah, I'm sending you on Skype. Just a moment. It's a export transport file video, and yeah. Then to listen to this video, you should have one software in your system. I don't know whether you have this or not, but yeah, this software. If not, you can just use this software to listen to that one. Okay. Okay, so this is about the transport file conversion. You just try with the C port, SIM port also, okay, as per the video, but this is enough. Converting using the export transport file engine is enough. Okay. Yeah, now you know what is a proc compare, right? So what is a proc compare? Proc compare means using this proc compare, you can compare the properties of a data set, or you can compare the number of observations and number of variables also you can compare. So compare the properties, properties, number of observations and number of variables. So for example, if you want to compare two variables, so we have two variables. Okay, for a variable we can get output about differences in. See, we have two variables. Then what the differences you can find in the variables? You can find the difference in the type, length, format, informant, and labels. Okay, you can compare both the data sets also. If you have two data sets, then you can compare these things when they are created, when they got last modified, the number of variables, number of observations. So these things we can compare. Okay, so for example, if you are comparing two observations, then the value of each record we can compare. Means each observation you can compare using this proc compare. So usually when we use this proc compare, it's very less used in the clinical. See, for example, the same type of data or the same type of work assigned to two SAS programmers, then they created the data set. Then if you want to compare both the data set, then they will do, and after comparing they will tell you like where the errors are. Then they will rectify whether the error happened in the one user data set or second user data set. So they will just compare the work. So usually using this compare, it's not possible to validate the huge work. Okay, it is just simple comparison we can do. Okay, let us see this example. We have one data set like this: unique, weight in kg, date of birth. See here how it is: 20, 30, 10, 02. Means they are not in a ascending order, right? See, 10, 20, 30, it will be like this, but they are not in a ascending order. Okay, so first see the contents. So what is the content is showing? We have three variables. Okay, so it is the contents, proc contents. Now yeah, here I am creating a new table. Okay, proc compare. So set, either you can use the set or you can use SQL. We are comparing a new table, creating a new table. What's the new table? Compare. Okay, means we have to have two data sets. One is compare, the first one is the demog and compare. Open the demog, it is same. When you open the compare also it is same, because we just made a copy. Okay, both are same here. Then as these two are both are exactly same. Okay, when both are exactly same, then you can use procedure here. Proc compare, base is demo, compare with the compare. Okay, submit it. Then a clean compare output. See, when they are modified, 1st January 14, 1st January 14, but we create it on 18 second and it is 59 second. Only this is difference when they are created. How many variables, how many observations, how many variables are in common, what's the first observation, everything is same. See, no unequal values were found, all values compared are exactly equal. Okay, now we are just sorting the first one by date of birth. When you sort, what happens? Then automatically here the date of birth is changed: 1931, 1934, 49, 49. But in our first data set we have 49 first. It's not in a sorted. Then when you sort in ascending order or some descending order, then automatically the content will get altered, right? Then you compare it. Proc compare demog. Okay, proc compare. Submit it. Now it will give you what's the first one, what's the last one. This comparison, right? You just submit it and check the output. Okay, so it is just to compare. Clear? So this is about the proc compare. It is just to compare the data sets or compare the variables or comparing two observations. Like that, you can use the comparison. Now you are talking about the MEDRA. Okay, it's a MEDRA means it's a medical dictionary for regulatory activities. Okay, so for MEDRA, first of all you should install the MEDRA dictionary. So I have one old trial version. I'm installing this in my system. So yeah, I'm just installing it. Okay, so we install this one, Medra. So this is the Medra. So it's showing Medra MSS4. First, let us see what's the Medra. Medra means it is a medical dictionary for regulatory activities. Okay, so there are different versions. Okay, about Medra. If you don't mind, just give me 2 minutes. I'll be back. Hello. Yeah, sorry, I'm back. Uh, okay, so we're talking about the MEDRA, right? So MEDRA means, it's a medical dictionary for regulatory activities. So usually there are different types of dictionaries, right? ICD, International Classification of Disease. Okay, International Classification of Disease, that is the one dictionary. Then we have some CPT also. CPT means current procedural terminology. I mean, like, US health insurance system, they use different, I mean, for insurance claims, okay, to, I mean, reimburse the physicians, they will use this type of the coding, medical coding we call it as. So that coding is different and this coding is different. There are so many like dictionaries are there, coding dictionaries. Co-start, WHO drug, Medra, like this we have so many dictionaries. So usually this Medra dictionary is recommended by the US FDA, because whenever somebody is writing, the diseases, so they should have the same verbatim words. See, usually what I do, I spell it as a fever. Some people they call the same as a elevated body temperature, and some people they call it as a pyrexia. Some people they call it as a hypo or hyperthermia. So there are so many names for a disease or some signs or symptoms, right? So if everybody is using their own name, then there won't be any uniformity in the data, right? See, for example, because of taking this drug, what are the adverse events you noticed? Then your report will be like this: 20 like subjects, reported fever. 20 like patients reported hyperpyrexia. Then 40 patients reported as elevated body temperature. So 40 patients reported as, body temperature is more, like that. So see, instead of saying that 100 patients are reported as some fever, so that will be good, right? So usually when the like people see the report, what they think, so because of this drug there are so many adverse events, right? They will think like that. So instead of that, if you use a common terminology, then adverse event is only one adverse event, that is a fever. So all the patients are reported as a fever. So there's only one contraindication, okay, for this one. Okay, so I mean, this is a small example, but there are so many disadvantages of using the verbatim words. So what we do, we use the common terminology. So what is the common terminology? So common terminology is there in the MEDRA. So all the CRO organizations or the clinical research organizations should use the same terminology as mentioned in the MEDRA. So this is about the signs, symptoms, and the signs, symptoms, the diseases, not the medicines. Okay, see, paracetamol, same meaning. Acetaminophen, same meaning. Okay, means the drug is same, but the drug is called with different names. So the uniformity in the drug name will be, see, the aspirin, and there is a separate name for the aspirin also, right? So sometimes they use the branded names, Disprin. So some people they use some like this. They use different brand names also in the place of medicine. They use different brand names also sometimes, right? They are famous with their brand names. So I mean, there is a separate dictionary called the WHO drug. WHO drug. So to code the, the medical, the medicine name, then we use a different dictionary, WHO drug. So here, this dictionary is for the disease signs and symptoms. Okay, but here again there are some levels in the MEDRA. What is the level? See, for example, because of this drug, 20 people got cold, 20 people got flu, then some 20 people got cold, cough, okay, respiratory tract infection. So we are giving different names, but all are related to the respiratory tract infection only, right? So though you use different terms, okay, then finally you are classifying it as a SOC only. Means, see, you got some itching, that's the adverse event. You got some scare, okay, on the skin, it's an adverse event. So you got some, the pus filled lesion in the skin, that's also, means all these are related to the skin only. So instead of giving different names, what we do finally, skin or dermatology related events are some 50 or 20 like this. So we will classify them into the system organ class. Like this, there are different levels in the MEDRA. Okay, let me show you what are the levels in the MEDRA. See, these are different levels. First is the lowest level term, preferred term, high level term, high level group term, system organ class. But usually in the clinicals we don't use this high level term and high level group term. We don't use this. Only we use the lowest level term, preferred term, and system organ class. Okay. And see, it's a MEDRA coding of the adverse event. So usually, adverse events only we will use the MEDRA coding. Okay. System organ class are 26 terms. High level group terms are 332. It's a version 6.1 I am talking about, but we don't know which version currently people are using. Yeah, current version is 17.1. Recently they updated in the September, because every 6 months they update. See, March, then September, March and September, March and September. Okay, so they have given understanding MEDRA. So if we can go through it, we'll understand what's the MEDRA. So just have a look at this website also. You'll understand what's in MEDRA. See this example. Preferred term is influenza. Influenza means viral upper respiratory tract infection is the highest level term. Then above to it, respiratory tract infection. Above to it, respiratory, thoracic and mediastinal disorders. So like this. Either you can classify the influenza as this one, or else you can classify influenza as infestations or infections. See, if it is a influenza viral infections or viral infection disorders, then infection means okay. I mean, if you are confusing with this, then I will explain with one more example also. Yeah, let us take this one. It's already coded one. Okay. So in the case report form, this is the adverse event experienced by one patient. Okay. What is the adverse event? Mild pain around the ulcer. So it's an adverse event term reported. Then you have to convert into lowest level term. Lowest level term is pain. Okay. Pain, then there is a code for the pain. Okay. Then when you go to preferred term also showing the pain only. Then what is the number for code? Pain, same number. And high level term, just bit up. What is up? It's a high level term is pain and discomfort. Then high level group term is general system disorders, NEC. Then later general disorders and administration site conditions. So this is the system organ class. Okay, for example, take the hyperglycemia. Okay, hyperglycemia is nothing but a sugar, right? Diabetes. More glucose content. Okay, hyperglycemia. So usually this is usually what we do in the normal we call it as a hyperglycemia. Then hyperglycemia means it is a, when you see the lowest level term, it's hyperglycemia only. Then hyperglycemic conditions, high level term. Then high level group term is glucose metabolism disorders including diabetes. Then finally it comes under metabolism and nutritional disorder, like this. So where these codes come means we have this dictionary. In this dictionary you have to find out, for example, eye disorders, congenital. Okay, like this. For example, a patient is having congenital lacrimal passage anomaly. Okay, so this is the lowest level term. So whatever you write here, then it will give the lowest level term. When you right click and open the details, then it will give the what is the lowest level term code. So it will be copying this code. And what is the highest level term for this one? Again, get the code. Then this is the highest level term. This is the highest level group term. Then system organ class. So like this, we will be doing the coding. Okay, but don't bother about this coding. Usually the data managers, they will do the coding. Okay, not the SAS programmer. Just for understanding, I explained you. But I mean, it's not like that. So if they ask you to do the coding also, you can do, but it's not like looking and doing the coding. Okay, there are different ways to do the coding. Okay, you can see a lot of information over the net like this. Medra and SAS. You see, there are so many papers out there because you can use the SAS formats as a Medra. So you'll be converting all the Medra formats into the SAS. See Medra data as SAS formats. So this is one paper which will explain you how to use the SAS to do the Medra coding. Okay, it's by the just coding only. Now we have one more paper also. Okay, Medra coding system, a SAS based Medra coding system. So they release different versions. Okay, it's a SAS based only. Okay, so how they will do? Okay, how they will do? First, Medra flat files. So Medra is there. Okay, then dataset to code. What dataset we are using the code. Then auto coding techniques are there. So we have to use the what we say, the particular version. We have different versions in the Metra. Okay, the desktop version is there. You know, these are different versions. See, it's a introductory guide Metra version. Then actually there are different versions. Desktop version is available, and some SAS related versions also available. So like this we have different versions are there. Okay, so anyway, don't bother about this one. If you can understand the concept, then that is enough. Okay, then what the SAS programmer role in the Medra? See, for example, data given to us like this. So this is the adverse event experienced by one patient, HED, headache, right? So some doctors they write HED, some doctors they write HED ACE, headache, headache. So different verbatim words are there. So instead of using these verbatim words, what we do, we convert using the Medra coding. We convert this into the common one. So this is the code, and this is the preferred term. So preferred term will be correct only. It won't be like this. Okay. So MI means myocardial infarction. Okay, broken leg. It is showing as the preferred term lower limb fracture. So we should use this verbatim. Instead of verbatim, we are converting into the this adverse event preferred term. Then after converting, then you submit it. We got the, see, this is the frequency we got before converting, because here we found the frequency of this AE verbatim. Okay, then it is showing, you know, these many diseases are there, 1 2 3 3 6 like this, but really these many diseases occurred? No, all these three are same, right? So we are doing the frequency of adverse event PT only. Now you got the like this. So because of this drug, these many events not happen, only these events happen, but the frequency of the headache is 3, myocardial infarction is 2, but here it is showing MI separately and myocardial infarction separately. Okay, so when you are doing some frequency, okay, then it's better you summarize the adverse events, only the preferred term, or someday they will give the SOC. So whatever mentioned in that SAP, so based on that we'll be doing this method. Got it? So this is the common clinical programming we'll be doing in our regular job. Okay, now this is one part, okay, the programming. Now next topic is we are talking about the C-disk, the C-disk implementation. So this is very important one, cdisk.org. So C-disk means it's a clinical data interchange standard consortium. So what is C-disk and what are the standards we have to learn? Okay, so we'll be discussing these topics in detail. So as a SAS programmer, you know this C-disk means it is all about the clinical data only. See, clinical data interchange standard consortium. It's not about how to conduct the clinical trial. It is about how to use the clinical data, okay, according to the standards. See, every organization, the clinical research organization, if they send the information to the FDA in different formats, let us assume we are sending in a different format, and some other company is sending in a different format, and some other company also they are sending that in a different format. So if everybody is sending in different formats, then it will be difficult for them to cross check all the reports and send the compliance report. So what we do, so if you are sending in the same like format, or if you are sending in the same standards, then it will be easy for them to cross check, right, and also send the reports in time. So all the CRO organizations and some countries, they combinedly made these guidelines. Okay, so there are some standards and implementation. So there are different standards, right from protocol creation to until sending the reports to the FDA. They developed so many guidelines. So as a SAS programmer, because we never involved in the data collection, so no need to bother about the lab standards and C- products. So as a SAS programmer, you should know the SDTM and ADAM standards. Anyway, to define the SDTM ADAM, you should know the define.xml. So I will be explaining you these three standards in detail: SDTM, ADAM, and the define.xml. Okay. So before going this, first what you do, click on this SDTM products. Okay. Then in the SDTM products, we just keep download all this. So we have, we'll be learning the SDTM implementation guide. So you just download this zip file, download a complete document package related to the SDTM. Okay, you just download and keep ready in your system. And also we download the ADAM also, because we will be learning this analysis data model and the SDTM. So we just download this. Okay, the complete analysis data model implementation guide and model also. Okay, download and keep ready in your system before tomorrow's class. And you download the define.xml also. Okay, we have the define.xml, you just download. So download these three, and also if possible you just download in the ADAM standards. So we have so many analysis data models like this. You download the time to event, adverse event analysis structure. You download some ADAM examples document also. Okay, so download and keep ready in your system before tomorrow's class. Okay, so tomorrow we'll be starting the SDTM. Okay, I don't know whether we can finish the SDTM tomorrow itself or not, because we have the code also. Okay, because the implementation also we should understand. It's not simply reading the SDTM guide. Okay, we should implement also. Got it? Yeah, so that's it for today. Yeah, tell me. Midra, see, actually Midra is the role of the data managers, right? So here I can't explain you very clearly about the Midra. You should know the hierarchies: preferred term, low level term, high level term, high level group term, system archon class. Okay, then which version we are using. Okay, then that's it, because I can't show you practically, because that is not a free version.
No, one question I have.
Yeah.
I just have one question. Like
Yeah.
now in phase 1, suppose you've used Medra 10 version 10. Phase 2 you've used Medra version 12. Phase 3 you've used Medra version 16.
Yeah, no.
How will you do a report with all these three phases
Yeah,
together?
that's a good question. But nothing, no changes in that version, just except adding some terms, that's it. Okay, because that Medra is a very recent one. Okay, see, when you see the WHO dictionaries, or when you see the CPTR, the ICD, those dictionaries are very sophisticated. But the Medra is not that much sophisticated, right? Medra is just a, recently it came. Okay, so when you see about this Medra, why they are upgrading once in 6 months, means still they have to add so many terms to it. So it's in the starting phase only. So that is the reason every 6 months they are increasing some terminology to it. That's it. But you won't find see, okay, let me tell you. See, this is the Medra desktop. I have this version 3, right? Okay, the look and feel will be same here. Just here you go and see it is showing Medra 16.1. Okay, click on it, then action, then automatically it will load from that website and it will be converted into 16 like this. Got it? So like this.
One more thing, how did you get the numbers? Where did you get the numbers? Like in your data set you had some numbers, right? Like
here see I disorders. Okay, so you just click on this term and right click, open the details, it will show. So this is the Medra code. This is a code and this is a term. Okay, Bhupal.
But now as a SAS programmer, we won't know that. No, we won't know. We may know only congenital glaucomas. So what do we do as a SAS programmer? Should we get to know, how do we get to know what exactly this is? How are you going to merge this with the AE data set?
No, that's
How
what
are you going to merge the meta codes?
That's what I'm saying, right? See, for example, okay, let me show you a small example. Here, for example, we have one. Yeah, let me show you. So when collecting the data from the case report form, right? So let us assume we collect in the case report form, the data is like this: hyperglycemia. Okay, hyperglycemia is there. So if you write this hyperglycemia, they won't accept. Okay, they will ask you to submit the report or make a table based on the SOC, system organ class. Okay, so in the MEDRA, what we do here, just to type the hyperglycemia and then click on the search. Okay, now it will give like this. Hyperglycemia means, okay, here it is showing select SOC to search where these are. So it is showing all the 26 system organ class. So where this hyperglycemia comes from, usually it will show here only. But the problem is this is the trial version, it contains no all the terms. But if you just type the hyperglycemia and click on the S, then automatically it will show here. System organ class, high level group term, high level term, it will show everything here like this. Then simply, for example, okay, I just copied, okay, in this, even it is not showing the verbatim words, that's the problem. Okay, let's type as a glaucoma. Now it is showing. Glaucoma means it is showing in two conditions, eye disorders. Okay, then right click here, open the details. Now you can see the code here. Then that code you write here. If it is a glaucoma,
where do we write? Do you merge that, or do you create a data set called AE codes and then you merge it?
what? You are just coding, right?
No. How you will write it? Like you will open an Excel file and you will start writing, you will start typing it, or is there something that you can merge
That's
this
what I'm saying.
MEDRA
Right?
with?
No. That's what I'm saying. There are different versions are available. Okay, so there is some SAS based coding is there. Okay. See, MEDRA and SAS. If you can see these papers, you will understand. Okay, a SAS based MEDRA coding system is there. Okay. So SAS based MEDRA coding is there means, if for example you selected one verbatim word, glaucoma is there, you just select that glaucoma, okay, right click it, automatically it will show the dictionary. Okay, it will show all the words. Okay, if you just click on okay, then that will be loaded into your Excel sheet like that. So they develop this automatically, but I don't have that. See, auto coding is there, auto coding. Okay, so I don't have that version. If I have, then it's very easy. Just to type it and click on it, automatically it will see. For example, fever. So if you type as FIVER, we are not typing FEVER. If you type as FIVER, because the doctors they write in a wrong way also, right? So if you type FIVER, then automatically it will suggest. Okay, it will suggest. If you, there is a right click. If you click on that one, then automatically it will show you FIVER. It's spelled as FEVER, and you click on the FEVER, then it will show all the codes as well as the high level term, high level group term, system organ, so everything it will show you. Okay, it automatically builds into the database. Okay. Anyway, the SAS programmer is not involved anyway here to do the MEDRA coding. Okay. So I don't know whether the videos are available or not, but I saw a video in the YouTube that how to do the MEDRA coding. Because if you have the version, then we can do it. 1 second, let me check.
When do they use WHO and when do they use the
WHO for the drugs.
Because both I've seen.
No, the WHO is drugs. I told you, right? Paracetamol, acetaminophen.
For concomitant medication, is it?
Yeah, for concomitant medication. This is for the adverse event.
So MEDRA is for adverse events and WHO is for drugs.
Concomitant medication. Yeah, see here we have 1 hour session it is, you know. Yeah, I think if you can listen to it, you will understand. It's almost one hour session.
Mitra coding, okay.
You can see here they use the Oracle Clinical. Okay, Oracle Clinical software, they use this Mitra for the Oracle Clinical. And this is the different one. Basics of coding and billing, this is a different one. Okay, just see only the Mitra, because this is a, the insurance system, this is. Okay. But the problem is, this video also, it's not showing you how to code. So they explained us like what is the Mitra, document and company standards, okay, in which phase we have to use. They explained us, some basic things about how the dictionary will be, what are the system organ classes, how to use the coding, when they started. So this type of history is there, but they didn't show any type of like usage. But anyway, no need to worry. If you know the hierarchy, what the terms we have, what is the sequence, okay, that is enough.
Okay.
Yeah, so this is about the Meta. Now, yeah, then tomorrow we'll meet and we'll start the SDTM, but please download the content, SDTM, ADAM, and the define XML, okay, before tomorrow's class. Okay, so tomorrow I will try to spend 2 hours. Okay. Okay then, thank you.
But please go a little slow. It becomes difficult to absorb everything together.
Okay, no. Anyway, tomorrow we'll be doing a bit slow only, because I